Welcome back to lecture 2, the process of science. So in lecture 1, we took a look at what is biology. We talked about what makes things alive. We talked about different domains. We talked about the different levels of hierarchy within living things. And in this lecture, we're going to talk about how we figure all of that stuff out, as well as the 5 unifying themes in biology. These 5 themes that you're going to be able to see throughout everything that we do for the rest of class. So again, we're still in chapter 1. Specifically, we are going to be looking at 1.4 to 1.14 in your book. So let's jump into that. So the first question we want to ask ourselves is, what is science? So science is a way of knowing about the world around us. It's an approach to understanding the natural world. Now, what's super important is that in science, we're not just making things up, right? We're not just looking around and making up nonsense about why things are like they are. There's a reason, there's a protocol, there's a process that we go through for it actually to be considered science. And the most important part of that is that science uses evidence-based processes. So we have to actually be able to test and show things through scientific experiments before we're able to make a claim about it. The scientific approach involves making observations, forming hypotheses, making predictions, and then testing those hypotheses via experiments and additional observations and the analysis of data. Now, when you have enough data and you've done enough experiments and you've tested a hypothesis enough times, so this involves a lot of data, this involves more than just one experiment, this involves a lot of different scientists doing the same experiment over and over and over and getting the same results. When that happens, we create what's called a theory. Now, in the general public, the idea of a theory is not really well understood because my dog is crying and laying under her leash, and I have this theory that she wants to go for a walk, right? Or I have a stomach ache and I have a theory that I might have eaten too many M&Ms the other night. We throw around this word theory for an idea, like I have a theory, I have a guess, right? Well, in science, that's not how we use the word theory. A theory is a really serious thing in science. Like in science, gravity is a theory. Right? The theory of gravity. And we all know that gravity exists, right? If you're questioning that, go jump off something very short so you don't actually hurt yourself. But when we've done a lot of experiments, we've tested the same hypothesis over and over and over, and we have a bunch of data that all suggests the same thing, that's a theory. Just like evolution is a theory. So evolution, just like gravity, is a thing. So that's what theory means in science. It's a thing. It's something we've shown over and over and over again. How do we do this? Now, you might remember from elementary school, you were probably taught the scientific method. It probably looked a little something like this, right? You might have even heard the Queen Rachel Hope's "Every coward gains courage." QRHECGC. Question, research, hypothesis, experiment, collect data, graph, conclusion, communicate results. You might have seen different versions of this, but it usually goes something like you make an observation and you ask a question about it. You do some research, you read, you watch videos, you ask other people, you try to get some background information to help you answer that question. Then you make a hypothesis, an educated guess about what's going on. Then you're going to make some kind of experiment where you're going to collect data in order to test the hypothesis you just made. Then you're going to graph it, and you're going to look for trends, and that's going to help you generate results. And from those results, you're going to draw a conclusion. You're going to reject, or you're going to fail to reject your hypothesis based on the data you just gathered, and then you're going to communicate your results. So you've probably seen some version of this before, and this is generally how science is done, but, right, here's a different method, looks pretty similar. But as a professional scientist, while this is generally how science happens, I want to caution you that there's actually a very big problem with the scientific method as presented in the 1, 2, 3, 4, 5, 6, 7, 8 process. What's the problem here? Science is messy. Science almost never happens in order, right? In that scientific method that you're taught in elementary school, if you get to step 4 and it doesn't work, what happens? You got to go back to step 1. Right? In science, the steps don't always happen in order. You can also repeat steps. Sometimes you repeat steps 100 times. So it makes science interesting, right? Science is messy. So while the scientific method is a good kind of overview of how we do science, don't take it as step-by-step instructions on how to do science, because every experiment I have ever done has been a little bit different. And that's okay. That's how it should be. That's how we figure things out. Scientists are actually very creative because they have to be. This is the best picture that I've ever seen of what science actually looks like. We ask questions, right? We make observations, we ask questions, we try to explain them. We investigate them, we analyze them, then we try to explain them again, we investigate again, we analyze again, and it keeps going and it keeps going and it keeps going. That's something about science, is it's never done. We are constantly asking questions, we're constantly trying to explain things, we're constantly investigating, we're constantly analyzing, because even if you answer your original question, it always leads to more questions. A scientist's work is never done. So the key to science is asking questions, asking questions, asking questions. Why? Now here's an interesting question for you. If that's what science really is, just constantly asking why, who are our largest group of scientists? Any parents out there? Children, right? Children are typically born with this great curiosity about the world around them, and they're constantly making observations and asking questions and testing the things around them. Toddlers are little scientists until we take that all away from them with standardized testing. But asking questions is the foundation of science, being curious about the world around you. So let's go through, though, a couple of the really important conventions of doing science, really important ways of how we do science. So at the core of every good experiment is a good hypothesis. So at its core, a hypothesis is an educated guess, right? We've made an observation, we've asked a question, hopefully we've done some research or we have some prior knowledge, and then we're going to make a hypothesis. Now, there are bad hypotheses and there are good hypotheses. In order to make a good hypothesis, that hypothesis must be clear and concise. I must be able to read your hypothesis and know exactly what you mean. A good hypothesis is made as a statement, not a question. It's testable and it's measurable. It includes cause and effect. So because of this, this is going to happen. If this happens, then this happens. Cause and effect. And it's repeatable. So other scientists could do that same experiment and get similar results. All right, so that's a good hypothesis. So let's take a look. An example of a bad hypothesis would be, will studying affect your grade? Right? Why is this a bad hypothesis? Now, it is fairly clear and concise. I know that you want to look at studying in your grade, but it's not made as a statement. This is a question. And it is testable, but we're not really sure what we would be testing for. Like, how much are you studying? How is it affecting your grade? So we have some level of cause and effect here, but we're not really quite sure. Like, are you saying that studying more or less will make your grade higher or lower? And because of that, it's not really repeatable because we're not exactly sure what you're asking. So let's take a look at the other example. Studying more will increase a student's chance of earning a higher grade. So that is clear and concise. I know exactly what you're asking. This one is made as a statement, and it is testable. We're going to compare the amount of time that you study. We could have one group of students who don't study anymore, in one group of students who do study more. And the effect, that's the cause, the amount that you've studied, the effect is the more you study, a higher grade. And it is repeatable because now we know exactly what we're looking at in this experiment. Hypotheses, like I mentioned, can be tested, and they're tested using controlled experiments. Experiments are very, very well thought out. And in experiments, we often manipulate one component in a system in order to observe how that affects change. So I mentioned cause and effect. So we're going to be manipulating the cause. In our last example, it was the amount of time you studied. So we could have one group, which we would call the control group, not do anything different. So they would study the same amount. The control group doesn't get changed. The other group, the experimental group, we're going to manipulate something. So in our last example, we would have them study more in order to test whether studying more would increase their grade. Now, the factor, the thing that we're manipulating, the thing that we're changing is what we call the independent variable. So in our example, the independent variable, that thing that we're changing or manipulating, is the amount of time that's studied. So in that experimental group, we're going to increase the amount of time that students are studying. And so the amount of time studied is our independent variable. And here's a hint. For the level of the class that we're taking right now, if you ever see time or anything that has to do with time, time is going to be your independent variable. When you get up to higher levels of science, that doesn't always hold true, but for what we're doing in this class, time will be your independent variable. Now, we're manipulating that independent variable, and we are trying to see if that's going to change the other variable, right? Cause and effect. It's going to affect the other variable. That other variable we call the dependent variable. So this is the variable that depends on the manipulated variable. So in our example, the dependent variable would be your grade. There are different types of data that we can collect. So we're going to move away from the example that we were just giving and talk about data more generally. We have quantitative data and we have qualitative data. Quantitative data is your counting data. So this is data that's represented by numbers. Qualitative data is descriptive data. So it's data that's described in words. So take a look at this. If you take a look at the top example here, qualitative data, again, that's descriptive data or data that's described in words. An example of that is on a scale from 1 to, oh my gosh, so cute. How cute is teacher's dog, right? And after doing a bunch of experiments, the results come back, oh my gosh, so cute, right? That's qualitative data. Fluffy, squishy, stinky, you know, oh my gosh, so fluffy, I might die. Qualitative data, data that we describe in words. The other side of that is quantitative data. That's our counting data that we express in numbers. So the number of students in a class, height, weight, etc. So those are our two types of data. So let's take a look. So here is an example of this graph where we are looking at the average change in heights of adolescents over time. So we have two variables. And I want to point out that for the level of science that we are going to be doing, you only want to be looking at two variables for your experiment. You want one independent variable, that variable that you're manipulating, and you want one dependent variable. Just that. Don't get any more complicated than that, because then you have to start trying to figure out, like, well, was it really the independent variable that was changing that dependent variable, or could one of those be affected this? Stick with just one independent variable, one dependent variable for each of the studies that you're going to be doing at a time. So in this example, what are the variables? Well, if you take a look, the variables would be age in years and height in centimeters. Which one is the independent variable? That would be, in this example, remember I gave you a hint earlier that if one of them is time, time's going to be your independent variable. So here it's going to be age in years, and our dependent variable is going to be height in centimeters. Now you can think about this. Is your age going to change your height, or is your height going to change your age. So do you get taller as you get older, or is the fact that you're getting taller changing how old you are, right? It's the fact that as you get older, that's going to make you taller, right? So your independent variable is going to be age. Your dependent variable is going to be height. Now we're going to cover how to make a good graph in lab, but it's something that is near and dear to my heart. I love a good graph. So we're going to take just a minute here to look at this graph, and I'm going to mention just a couple of things about making a good graph. The first thing is notice the title. Please title your graphs, title your graphs, title your graphs, or I will sing about it. Title your graphs, all right? The title of the graph should tell me exactly what I'm looking at. A lot of the papers that I read are 45 pages long. They have 20 different tables and graphs, and I am not reading through all of them to find the graph that I want. So you better have a good title that tells me exactly what this graph is about. It should be short, it should be concise, but it should be descriptive. It should tell me exactly what I'm going to be looking at. Your independent variable goes on the bottom. That's what we call the x-axis. So down here at the bottom where we see age and years, that is the x-axis. That is where our independent variable goes. And then the side on the left is called our y-axis. That is where the dependent variable goes. Notice we have labeled each of the axes. So we're going to put height, our dependent variable, here on the y-axis. We're actually going to write out height. And then we're going to put our independent variable down here, and we're actually going to write out age. Notice we also have our units, right? Because age in years is way different than age in days or age in seconds. So always put your units, height in centimeters. And then you'll also notice that we space our numbers evenly. So 10, 11, 12, 13, 14, 15, 16, 17. We're not going to go 10, 15, 55, 142, 143, 107, right? We're going to space them out evenly. It doesn't matter how you do this. I mean, it matters how you do this, but you get to pick how you do it. So it could be 1, 2, 3, 4, 5, 5, 10, 15, 20, 50, 100, 100, 200, 1,000, 2,000. You have to pick which intervals make the most sense for your data, but those intervals have to be even on that same axis. Now notice though that your x-axis and your y-axis don't have to use the same intervals. So while we're going by ones on the x-axis, we're going by fives on the y-axis. That's perfectly fine, as long as if you're going by ones on the x-axis, you go by ones the whole way along that x-axis. And if you're going by fives on the y-axis, you go by fives the whole way on the y-axis. You also, one more thing about this good graph is you want to have your data take up the whole graph. The first time you graph, you're probably going to have your data in one itty bitty little corner of your graph, which is fine. We're going to practice it throughout the lab, but your goal is to have your data take up that whole graph so you're able to actually see the data a little bit easier. Because that's the whole point of making graphs, is it's really hard for us to just look at a whole bunch of numbers and see trends. Graphs, though, help us to see trends. So here's an example. Why doesn't a flashlight work, right? The power goes out, you grab a flashlight, you turn it on, and it doesn't work. So how do we figure this out? How do we science this? So the first thing is we made an observation, right? Our flashlight didn't work. Then we asked ourselves a question. Why doesn't the flashlight work? So now we're going to make a hypothesis. In this case, we're actually going to make two hypotheses. Hypothesis number one, well, maybe the flashlight doesn't work because the batteries are dead. Hypothesis number two, or maybe the flashlight doesn't work because the bulb is burned out. All right, so then we're going to test it. We're going to make a prediction, right? Replacing the batteries will fix the problem, or replacing the bulb will fix the problem. Then we're going to test it. So for hypothesis number one, we're going to replace the batteries. Then we're going to turn it on, see if it works. For hypothesis number two, we're going to replace the bulb, turn it on, see if it works. And if you replace the batteries and the flashlight doesn't work, then we're going to reject that hypothesis. If it does work, then we will, what we call fail to reject, which is kind of like accept our hypothesis. And we'll do the same thing for the bulb burned out. Now, a lot of times I get asked, I'm not a biology major, why do I have to take this class? right? Which is exactly what we're trying to answer with our science in our everyday lives discussions. But it's because we do these types of experiments all the time. We actually do science all the time and we might not even realize it. Think about the last time that you ran to check something on your phone and your phone didn't turn on. And what did you do, right? First, you probably panicked, but after you panicked, what did you do? Well, you made an observation. My phone didn't turn on. Then you asked a question. Why doesn't my phone turn on? Then you formed a hypothesis. Maybe the battery's dead. Then you tested that hypothesis. You plugged it in, you let it charge, and then you got results. Your phone turned on. So you drew the conclusion, well, the battery's probably dead, right? Science. We do science all the time and we don't even realize it. But you do this process of science all the time. So you're doing it. So let's look at an example of this in real life, because we can do this in the field. And that's what a lot of my actual research is, this type of field research. And I did this animal behavior research quite a bit. So we're gonna look in this real example of scientific process in action at these two different populations of mice. So there is one type of mouse that lives at the beach. And most of these mice that live at the beach have a really light color to their fur, where there's another population of the same species. So this is the same species of mouse, but one of the populations lives more inland in an area that's more brown. And you notice that most of the mice there actually have a brown coat. So there's your observation. You've noticed that most of the mice that live at the beach in the sand have a white coat, and most of the mice that live inland where it's brown have a brown coat. So then you ask yourself a question. Why do they have different color coats? Is that related to the environment? So then you're going to make a hypothesis. Remember, our question is not our hypothesis. Our hypothesis is a statement, and we're going to make a hypothesis. Something like the color of the mouse's coat helps them blend into their environment. Or mice that live at the beach will have white coats because it helps them to camouflage from predators. Mice that live inland will have brown coats because it helps them camouflage from predators, something like that. So then we're going to test it. So the way that we're going to test it is we're going to have our control group, and our control groups are going to be mice from their own area. So we're going to take the light colored mice and we're going to keep them at the beach, and we're going to have the brown colored mice and we're going to keep them inland. Our experimental groups, though, we're actually going to take some of the mice and we're going to switch them. So we're going to take some of the brown mice and put them at the beach, and we're going to take some of the white mice and we're going to put them in the brown area, and we're going to see what happens. We're going to see if there actually is a difference in the level of predation. So this is a real experiment that was done. And so if you take a look, the camouflaged models are the ones that came from that area. So for the beach, that would be the ones with the light coats. So you'll notice this is number of attacks. So you'll notice that the mice that were taken out of their environment and put in the other one, so the brown mice on the sand or the white mice in the inland area, actually had a higher level of predation. And we can probably guess that's because they stood out more, right? But we're able to show that with science. We're actually able to show that the mice that you transplanted from one area to the other were eaten. There was a higher level of predation among those. So there's a little science in action for you. So that's how we science. That's a basic overview of how we do science. And you're going to be doing a lot of that in lab, where we're going to get more in depth with how you science throughout the semester. But for the end of this lecture now, we're going to be looking at 5 unifying themes in biology. So these are going to be 5 themes that you're going to see throughout our study of biology. Theme number 1 is that evolution is the core theme of biology. I mentioned earlier, evolution is a theory in science. That means it's a thing, right? Theme number 2, life depends on the flow of information. We're going to talk about DNA here in just a minute. Theme number 3, structure and function are related. Theme 4, life depends on the transfer and transformation of energy and matter. Things need energy. Theme number 5, life depends on interactions within and between systems. So let's take a brief look at each of these themes. For a deeper dive, make sure you read the end of chapter 1. All right, so theme number 1, evolution is the core theme of biology. Evolution explains the unity and the diversity of all life on Earth. So evolution can be defined as change over time. So it's a process. This is going to be really important because you've probably also heard of natural selection. They're not the same thing. Evolution is the process of change over time. Natural selection is going to be the mechanism by which evolution happens. How do we know this? How do we know that evolution is a thing, right? We can't look back billions of years, except actually we kind of can. We can use the fossil record to show that life has been evolving for billions of years. We can look for patterns of ancestry. We can also use DNA. We've gotten a little more sophisticated now with our technology, so we can look at DNA to help tell evolution. But we have a lot of evidence that evolution is a thing. Charles Darwin, though, so we've known that evolution is a thing actually for quite a while. We've known that things are changing over time for quite a while, but we didn't really know how it was happening until fairly recently. And the man who's famous for explaining how evolution happens is Charles Darwin. If you're ever having trouble sleeping and you need something to put yourself to sleep, read his book on the origin of species. It's incredibly insightful and fairly boring. And it actually never mentions natural solution. He describes natural solutions. He describes natural selection, but he doesn't actually name it. It wasn't until later that we named it natural selection, but he describes the idea of natural selection. So again, evolution is the process of change over time. Natural selection, this idea that Darwin described, is the mechanism by which evolution is happening. So, mechanism of evolution. I'm going to give you one quick example, although we'll get into this in much more detail in later chapters. There's this very famous example of the peppered moth. This is a moth that lived in England during the Industrial Revolution. And if you take a look back here, before the Industrial Revolution, there were a lot of these really light colored trees. The bark was almost white. Now there's two different variations of this moth. There's a light version and there's a dark version. And before the Industrial Revolution, well, I actually want you to guess, which one of these do you think was more prevalent? Do you think we would see the light version or the dark version more? And I'll give you a little hint. These are eaten by birds. So think, if you were a bird, which one of these moths would you eat? Well, the answer is you would typically eat the dark ones because they're easier to spot, right? These light colored ones blend in, they camouflage with the light bark on the trees. And so we would find many, many more of the light colored moths compared to the dark colored moth, just because the dark ones stood out more and the birds would eat them. Well, after the Industrial Revolution, it actually stained the bark of the trees a darker color. So now take a look post-Industrial Revolution. Now, which one would you eat if you were a bird, right? Well, now the light colored moths stand out more. So the birds were actually eating the light colored moths more, and the darker colored ones were reproducing. And so now we were finding more of the dark colored moths than we were of the light colored moths, right? Whichever one the bird eats is not going to be able to reproduce. So it's the other ones that reproduce, so we see more of them. That's the basic idea of natural selection, is that some organisms are by chance going to have adaptations that help them survive and reproduce more than other organisms. So whatever adaptations they had, they'll pass on to their offspring. So we're going to see more of that particular adaptation in the next generation. That's natural selection. All right, theme number 2, life depends on the flow of information. I've mentioned DNA a bunch, and that's because that's what we're talking about here. The flow of information, what tells everything to do what it does is your DNA. The unity of life is based on DNA and a common genetic code. So what is DNA? DNA, deoxyribonucleic acid, is composed of these 4 nitrogenous bases. So adenine, guanine, thymine, cytosine. Adenine bonds with thymine, AT, think of at, A to T, and then guanine binds with cytosine, and they form this double helix. The order in which these nitrogenous bases appear in your DNA makes your genes. It tells your cells what to do. It tells your whole body what to do. Every single cell in your body has DNA, and the order of those nitrogenous bases in your DNA tells your cells what to do. So DNA is a genetic code of every living thing in the world. We're going to get into that a lot more in later chapters. Theme number 3, structure and function are related. So take a look at this cute little red panda, and if you take a look, it has this false thumb. Why does it have this false thumb? Right? Why? What is the point? Is it just there for funsies, or does it do something? Well, in life, usually nothing is done for funsies because it takes energy, right? It takes energy to make that little false thumb. So we're going to do it for a reason. And the reason, if you take a look, is to grab things. You know, take a look at your feet. Why are they big? Why do you have toes? It's for walking, right? Structure is related to function for pretty much everything at every one of those different levels of hierarchy. Theme number 4, life depends on the transfer and transformation of energy in matter. In other words, all living things need to get energy somehow. How do humans get energy? We eat tasty tacos, right? We eat. Not everything is going to get energy the same way, right? Plants don't eat like we think of eating. Plants aren't going to eat tacos, but they are going to be getting energy from somewhere, and they're going to be transforming that energy to do work. So if we take a look, this is the energy flow pyramid. If you ever went to 6th grade camp, this was one of your lessons. I used to teach at 6th grade camp, so it's one of my fondest memories. But almost all of the energy on Earth comes from the sun. There's some debate about places like hydrothermal vents and if it actually comes from chemicals, but really all energy on Earth at some point is tied back to the sun. So the sun's going to be providing our basic energy. But as humans, right, we can't just go out and eat sunlight. Something has to happen to it first. So the group of organisms that can use the sun's energy and turn it into food are called producers. Back in the olden days, you might have heard this level referred to as plants, but more than just plants are able to do this. This process is called photosynthesis. You might have heard of it. We're going to get to know it and love it in later lectures. But more than just plants do photosynthesis. There are some algae, some plankton that can do photosynthesis as well. So that base are called producers. The next level up used to be called herbivores, or plant eaters. Then we would have had omnivores that eat both animals and plants. And then we would have had carnivores, which just eat meat. We're moving away from that model now, because really what this pyramid means is the base level is the group of organisms that can turn the sun's light into energy. The next level up is what eats the producers. The next level up is what eats them. The next level up is what eats them, right? So there could be more than just herbivores, omnivores, carnivores. There can be more than 3 levels there. Also, sometimes you might have an omnivore eating a plant. So what we do now is the basic level here at the bottom is producers. The next level up are primary consumers, secondary consumers, tertiary consumers, quaternary consumers, and we can keep going with words that I don't know. But you'll notice a couple of things. So the primary consumers eat producers, secondary consumers eat the primary consumers, tertiary consumers eat the secondary consumers, so on and so forth. You'll also notice this is in the shape of a pyramid. Why? Well, that is because most of the energy is going to be going to that bottom level. That also means its population is going to be the biggest. Take a look around. What do you see the most of? Probably producers, right? You're going to see a whole bunch of plants. You're not going to see that many carnivores. Here's why. Plants are going to take the sun's light, turn it into food, right? What's it going to do with that energy? Well, the plants are going to use it. They're going to use it, and they're actually going to lose about 90% as heat, and only 10% of that energy is going to be passed on to primary consumers. So their population is going to be smaller because there's just not as much energy available to them. Primary consumers are going to do the same thing. They're going to use it and lose it. They're going to lose 90%, and they're only going to pass 10% of the 10% that they got on to the secondary consumers. So now once we're to secondary consumers, there's only 1% of that original energy left. So since there's not as much energy available, their population sizes are even smaller. That's going to go on for each of what we call trophic levels. So each one of these levels is called a trophic level. And at each level, 90% of energy is going to be lost as heat. Only 10% of the energy is going to be passed on to the next trophic level. So as less and less energy becomes available, population sizes are going to get smaller. That's why we see a lot of plants. We don't see very many carnivores because there's just not as much energy available to them. Once those things die, they're going to be decomposed, and then they're going to go through a cycle where their decomposing bodies are going to be turned into nutrients that are put back into the soil from which the producers grow. So it's this whole circle of life. And theme number 5, life depends on interactions within and between systems. So we actually talked about the 2 main properties of this theme in the last lecture. We talked about emergent properties. If you remember when we were going through the hierarchy of life, at each level, there's going to be a new property that was not present in the levels before it. It's emergent properties. And when all of those different things interact, that makes what we call systems biology, where we're studying all of those different interactions. And that's going to be really one of the core themes in biology, is looking at how all these different pieces are going to be interacting with each other and what the effect of that is going to be on life overall. All right, so let's review. What is science? Science is a way of knowing what's going on in the world around you, but it's a way of knowing that is scientific, right? Where there are some rules that we follow. In studying nature, scientists are going to be making observations, and they're going to form and test hypotheses using experiments and using data. A good hypothesis is clear and concise. It's made as a statement. It's testable. It's measurable. It includes cause and effect, and it's repeatable. Hypotheses can be tested using controlled experiments. These experiments can include qualitative or quantitative data, and they're going to include an independent variable and a dependent variable. There are also 5 unifying themes in biology. Evolution is going to be the core theme of biology. And then we also have life, which depends on the flow of information, known as DNA. Structure and function are related. Life depends on the transfer and transformation of energy and matter, and life depends on the interactions within and between systems. So that's the end of lecture 2 for week 1 in Biology 107. If it's not Wednesday yet, remember to hop on that Zoom call for our science and our everyday lives discussions that takes place every Wednesday from 6 to 6:55 p.m. via Zoom. Also remember that you are going to have a module quiz that is due this upcoming Sunday. And you'll also have a lab module that'll come after this. So take a look through this lecture module, make sure you get all the different pieces done, move on to the lab module, make sure you get all those pieces done, and then I will see you in week 2.